Good morning. Good morning and welcome. I'm glad you all could be here. Most of you know me. For those who don't, I'm Dr. Stephen B. Korn, and I'm most enthused about today's presentation, and of course our guest speaker and lecturer. My excitement and enthusiasm really go to the heart and soul of academic medicine. Today's presentation, I think, is a really fine example of what I believe academic medicine should be all about. It's the bringing together of people near and far to share ideas and research in an effort to extend what we know and to foster further thinking. So all of this is with the ultimate goal of improving patient care. So following the presentation, with this in mind, I've arranged for a 2-hour luncheon where we can informally meet with our distinguished presenter and spend some time together and share ideas. I'm confident that not only will we learn much during the time with him, but we also will have a two-way exchange where the excellent research and clinical work being done here may impact future studies and additional advances in the field. So today's guest speaker is Dr. Raphael Mechoulam. We'll provide some background and list some of his accomplishments. I'm not going to list all of his accomplishments because that's all we'll be doing today. We won't have time for anything else. In our correspondences back and forth, and he's been most gracious, I just want to quote. He said, "You really do not have to waste time with my honors and honorary doctorates." Well, let me waste a little time because it's really impressive. So Dr. Mechoulam is the most highly honored, respected, and awarded chemist. He's published more than 400 scientific articles. Early in his career, he gained prominence for the isolation, structure elucidation, and total synthesis of THC. I'm just going to advance our slides. It's easier to see it than to say it. So that's THC, the first one. And then later he reported the isolation and the identification of 2 endogenous cannabinoids. Over the last 50 years, Dr. Mechoulam's research has focused on chemistry, the isolation and synthesis of plant cannabinoids, biochemistry, in terms of cannabinoid metabolism, and chemical biology, the identification of the endocannabinoids and their effects. Dr. Mechoulam has received many honors, including honorary doctorates from Spain and the United States. In 2000, that's slide 1, there's another one. In 2000, the Prime Minister of Israel presented Dr. Mechoulam with the most distinguished Israel Prize. He is the recipient of the European College of Phycopharmacology Lifetime Achievement Award in 2006. In 2011, Dr. Mechoulam received the National Institute of Health Lifetime Achievement Award presented by the head of the National Institute for Drug Abuse. In 2012, he received the Rothschild Prize in Chemical Sciences and Physical Sciences. A few more. He is a former rector of the university and formerly headed the Science Division of the Israel Academy of Sciences. Dr. Mechoulam is currently with the Institute for Drug Research and on the faculty of medicine at the Hebrew University in Jerusalem. Dr. Mechoulam and collaborators are now also focusing on clinical research on THC in post-traumatic stress disorder and on cannabidiol in the epilepsy and graft-versus-host diseases. Research plans include exploring the anxiolytic antidepressant activities of cannabidiol in borderline personality disorders. I'm going to leave it there. There's so much more to tell. I'm going to let it come in his own words. So thank you so much for coming.
Many thanks for your warm words. I really appreciate it. It's always a pleasure to be in Boston. We spent a few months, many years ago, and we enjoyed every minute. And I'm looking forward to the next few days that we're going to be here. I'll start my lecture, this is the title. I start my lecture with a few commercials that I've been told that we have to have them. Well, then I'll move to a paper, to a review that was published not just a few months ago by 2 very distinguished scientists, MDs actually, both of them, at NIH. One of them is head of research at one of the NIH Institute. And there he says, as you can see, in almost all diseases affecting humans, the endocannabinoid activity is central. Now, this is a very strong statement. I've never seen any statement of this sort for any other system. Not for the dopamine, not for the serotonin, nothing of this sort. Now, that's a very strong statement. And I'll devote most of my time trying to show that maybe, just maybe, Dr. Kunos and Dr. Pahar are right, that there is involvement of the endocannabinoid system in many of our diseases, and hopefully we can go and use that involvement in order to help patients with these diseases. So let me go back 3,000 or 4,000 years ago. Cannabis has been known for a huge number of years. As a matter of fact, we still have the Assyrians who left us clay tablets, and so we know that they used it, and they say in those clay tablets, amongst many other drugs, amongst many other things, that they used cannabis, but cannabis, they do not use the name of the plant, they use the name for the effects. And there are three types of effects. One of them is what today we call the high, and they had a much better definition. They called it gamzigunu, and that's the drug that takes away the mind. Then they use it as a therapeutic agent, and it is apparently has to do with neurological diseases, and they use it also in religious rites. Then we can move forward about 3,000 years, and Dr. Reynolds, Russell Reynolds, he was the physician of Queen Victoria, and apparently Queen Victoria had migraines, or what he says, migraines in English, and he found that cannabis brought from India, not the cannabis grown in Europe, but cannabis brought from India, and we know the reason because it contains a different set of constituents, helped Queen Victoria. So we have a lot of use over the ages of cannabis of various sorts. But surprisingly, when in the 60s, about 50 years ago, I came back from the States. I had been here on a postdoc at the Rockefeller Institute, and I was looking for interesting topics. I was kind of amazed to find out that although morphine had been isolated from opium 100 years previously, and cocaine was known, of course, had been known for about 100 years, the active compound in cannabis had not been isolated in pure form. The structure, obviously, was not known. And we, of course, know that researchers and clinicians are not happy working with mixtures. Mixtures lead to a result that cannot be reproduced. Mixtures are not something that people can cite or go ahead with. And therefore, there was very little biochemical, pharmacological, and almost no real clinical work. That was about 50 years ago. So we thought that maybe it's a good idea to try to clarify the chemistry first. And using methods that at that time were modern, started isolating the material, and we were able to isolate about half a dozen or maybe a little bit more of compounds, all of which had more or less the same structure. Those of you who may recall your high school chemistry will see that all of them are more or less the same. They have this part, which is an aromatic structure. All of them have the same. And this part, which is a terpenoid structure, all of them have both. But the only compound which showed the activity for which cannabis is well known, the high, it was just, surprisingly, just one compound. And this is the tetrahydrocannabinol, or the THC. This is the only compound which caused the high. Of course, we couldn't test it in humans. We tested it in monkeys. And the other compound that was present in high amounts was this one, cannabidiol, which is not psychoactive. It doesn't cause anything, at least not in the monkeys at that time. But although they're very closely related chemically, they're almost the same, except that in this case, here, there is a ring caused by ring closure here, and this one does not have it, and this one has it. And This one is the active compound. And essentially, in monkeys at least, it caused all the effects that we know about. That was a little bit strange, but well, I'll tell you a little bit more later. We tested it in monkeys. This is a rhesus monkey that has not been given the THC, and it's rather aggressive. This monkey here has been given 3 milligrams per kilo of THC, and it doesn't sleep. It's kind of sleepy, and my students usually tell me that it takes up a professorial attitude. I mean, well, I don't know whether he's going to be a professor or not, but we found that this compound is the only one. We tested all of them, all the major ones. None of them caused anything in the monkeys. Now, for the next 20 years, we and many other groups, because the topic became of interest, looked at the metabolism, looked at the physiological effect, in some cases the clinical effects, and the one thing that was not well clarified was how does it act in the body. We know that most of the compounds that we are well acquainted with, dopamine, serotonin, opiates, and so on, they work on receptors, or in some cases on other binding sites, and sometimes on enzymes. And THC didn't seem to fit. A very important group in London, in Cambridge, in the UK, wrote that it acts not specifically on nerves. Now, that sounded strange, because if it acts on nerves, then all the others should do the same. They're almost the same structurally. Besides, they said these two compounds, the plus THC and the minus THC, act more or less the same. Now, for those of you who, again, do not remember their high school chemistry, compounds that have asymmetric centers are like mirror images. But in the body, only one of them normally acts, because essentially all compounds or all entities in the body, in the human body, in the animal body, have a certain stereochemistry. These two compounds have different stereochemistry, and therefore only one should act. In this case, both acted, and it did some strange, and people said, well, THC doesn't act specifically, and therefore it's not binding to a receptor and anything else. Well, it turned out to be all wrong. When we synthesized both this compound, which is not the natural one, and we compared it to the natural one, we saw that this compound is essentially not active. People had worked on the wrong mixture, or a compound that was not pure. So at this stage, we knew that THC acts on something which is specific, which is there not just because the compound acts non-specifically. And indeed, a group in the US, Alan Howlett, found that there is a receptor site. And a little bit later, a second receptor site was found. So today we know that there are two receptors. The CB1, stimulation of CB1 causes the high, the ganzi-gunu of the Assyrians. And the other one doesn't cause anything at all, actually. It works only in the body when there is a disease around. It seems to be a protective mechanism of a sort. Now I'll be speaking a little bit more about it, because that seems to be an important novel therapeutic site, a receptor which is almost not there at any time, certainly not in the central nervous system. It's mostly in the immune system, but at very low levels. In the brain, it pops up when there is a disease. Otherwise, one doesn't see it. In any case, we try to find out what happens. I mean, what are the mechanisms? And Alan Howlett, that found the receptor, found that the receptor is present in all those places in the brain, areas of the brain, where one should expect it to be because of the actions of cannabis, of THC, all the areas, and here the basic ganglia, globus pallidus, and so on and so forth, which cause all the effects that we know about, the movement control, the coordination of the body, learning and memory, higher cognitive functions, and so on. So here we have something, a receptor that is to be blamed for the activities of cannabis. THC acts on this particular receptor. Now, we don't have receptors in the body to be activated by a plant out there. It doesn't work that way. We have receptors in the body because these receptors are usually, always as a matter of fact, stimulated by something that we produce, an endogenous compound that we have. So the next step was, let's look for these compounds. I wouldn't go into the details. There was a lot of work, a lot of time that we invested. These, and obviously new neurotransmitters are found in extremely low amounts, and we were lucky that we thought that we should be looking for a compound which is different from THC, but it should have the same physical properties. And THC is a lipid-soluble compound, so we didn't look for water-soluble compounds or peptides. We were looking for a lipid-soluble compound, and indeed, after quite a lot of work, we found that there are two compounds. This one was the first one that we isolated in 1992, and this was isolated a couple of years later. This was from the brain, and this was from the periphery. And these two compounds are completely different structurally from THC, completely, except for their physical properties. They are lipid-soluble to the same extent as THC, and they bind, both anandamide and 2-AG, bind to the receptors to the same extent. There is one difference, and it is normal for the body. Endogenous compounds of this type are normally broken down when, after not a very long period of time. THC is not broken down so immediately. It takes a couple of hours. So the activity of THC goes on and on, while the activity of anandamide and 2-AG is a short-term activity, and the body then breaks it down. Now, this paper brought a lot of interest at the cannabinoid system. As a matter of fact, the paper on anandamide has been cited about 3,500 times in the literature, which is not a bad citation rate. And people have found that this is, as I said, involved in many, many different activities. And there is a huge amount of work, and NIH, which supported my work, has supported my work for the last 45 years, are extremely interested. They're spending a lot of time and money on the endocannabinoid system. I don't have the time to go into great detail on the endocannabinoid system, but it involves the two receptors, the CB1 and the CB2. It involves the two compounds which we discovered in the 90s, anandamide and 2-AG. It involves the enzymes that synthesize these compounds and the enzymes that hydrolyze these compounds, as contrary to many of the other neurotransmitters, which are synthesized and kept to be released when needed, dopamine, for example. Anandamide is formed when and where needed. If, for example, there is pain, it will be formed around in the area of the brain that has to do with pain, not all over. It is formed there on demand, and then it is broken down. And people have thought that maybe they can use the enzymes that synthesize these endogenous cannabinoids and the enzymes that break down these endogenous cannabinoids in order to modify the levels of anandamide and 2-AG, and thus use them as potential drugs. Before I go on with these things, I'm a bit frustrated here. When people discovered insulin in the 20s, within 6 months it was a drug. When corticosteroids were discovered in the late 30s, early 40s, it was a drug within 2 years. And the companies have made good profits on these drugs. Anandamide and 2-AG have never been given to a human being. And the reason is that somebody has today, under the conditions that exist today, under the rules that exist today, one has to test to do toxicology in a very, very wide spectrum of activities, and that leads to, and that costs $30,000, $40,000, $50,000, and companies will not do that because they cannot patent these compounds, and therefore these compounds have never been given to human beings. There's a huge amount of work on animals, there's a huge amount of work on in vitro, never on humans with these two compounds. So companies are trying, quite a few of them, essentially all the major ones, are trying to play with the synthesis and hydrolysis of these compounds, but not with the compounds themselves. Now, there are two other topics that are involved in the endocannabinoid system. One of them is compounds that are related structurally to anandamide. Anandamide is a compound formed from a fatty acid bound to amino acid. Fatty acids and amino acids are prevalent in the body, and there are quite a few of them in the body, possibly. There are about 200 or 300 of them. After we published anandamide, people looked at are there other compounds present, and they found about between 200 and 300 compounds of this type. To the best of my knowledge, none of them binds to the receptor, but the very few of them that have been looked into, and we have looked into a few of them, they definitely have a lot of activities. And so here we have a rich possibility to look at compounds which obviously have something to do with the body. I cannot imagine that the body will spend so much energy and time, if you wish, to synthesize so many compounds just for the fun of it. It doesn't make sense. So chances are that we have there something of real importance, and time allows, I'll go and go into it again. Cannabidiol is one of the compounds found in cannabis, as I showed you before, and although it doesn't bind to the cannabis receptor, some of its activities can be blocked by antagonists to the cannabinoid receptors. So here we have something that goes downstream. It's associated, but we do not know exactly where and when. So people have looked, as I said, the endocannabinoid system. They have found that it is has to do with, here is a list of conditions: anxiety, appetite, immune system, memory, movement, stress. Cannabinoids have been shown to be very much involved in all these conditions. And for those of you who are interested in mechanism, the agonists, the compounds that bind to the receptor, that activate the receptor, they do things. The receptor is a G protein receptor, like many, many others, and it lowers the adenylyl cyclase in the body, and this causes blocking of calcium, entrance calcium release. On the other hand, it causes enhancement of potassium, and these effects have very important, are very important in the body in many, many respects. And there is another thing which is unique for the cannabinoid system. Most receptors are formed on the presynapse in the nerves. And then when these compounds are released from the presynapse, they move to the postsynapse and start a series of reactions. This is true for essentially all the transmitters, all the neurotransmitters. The cannabinoids have to be different. As I said, they are not formed and stay there. They are formed on demand, but they are formed on the postsynapse when needed. They move to the presynapse, and they regulate the release of the other neurotransmitters. One of their actions is regulation of many other transmitters. So they act on dopamine, they act on serotonin, and for some strange reason, dopamine is involved in quite a few diseases, from Parkinson's to schizophrenia. So obviously, the endocannabinoid system is therefore also involved in many other diseases. So here we have deep involvement of the cannabinoid system, of the endocannabinoids, with essentially all other neurotransmitters. Now, I will try to say a few things about the actions of the various types of compounds, and then I'll summarize. THC, the psychoactive compounds, has been found to have a lot of activities. I'll mention just two. One of them is appetite and feeding. Definitely THC enhances appetite, and blocking of the endocannabinoid system of THC blocks appetite. So we looked at animals that were just born, pups of mice, and we blocked the system, and these newly born pups refused to eat. I mean, the mother was there was a lot of milk, and they just didn't know how to do that. If one blocks their endocannabinoid system, they don't suck. They sit around and die of hunger. But if in the middle of, over the next few days, one gives them a lot of THC and release, and this causes the inhibitor to be kicked out, they start sucking again. So it is deeply involved. So a company decided, well, if this is the case, let's synthesize an antagonist to the CB1, and then we'll treat obesity, which is an important disease, if you wish, in the US and everywhere else, and we'll have a good anti-obesity drug. And they spent $1 billion developing an anti-obesity drugs, and it was released in Europe, not in the US, and it was there for about 2 months, and then it was withdrawn, because they should have realized that THC, or the CB1 receptor, acts not on obesity, it acts on many other things. For example, it lowers anxiety. So if you block the CB1 receptor, you not only block appetite, you also cause anxiety, because the CB1 receptor has to do with anxiety. So there were a few cases, people that wanted to commit suicide, and they immediately took it out, and the head of research at that time was told to go home. Now, another thing about CB1, we tried THC in vomiting and nausea in children that were undergoing cancer chemotherapy. Fortunately, as you know, children also have cancers. And in one of the hospitals, in Jerusalem, there is a department of pediatric oncology, and they were treated, and most of the children get well afterwards. But in the meantime, they vomit and they have nausea, and it's pretty nasty situations for the children and for the families. And we decided to try THC because it was known to block nausea and vomiting. And we started giving these children 5 milligrams of THC, more or less depending on the weight and so on, and the age, 2 or 3 times a day under the tongue in olive oil. And it was supposed to be a double-blind experiment. The physician came over and told me, no, I'm not going to continue with the double-blind because I know now who's taking it, who's not. Those that were taking it stopped vomiting immediately, nothing. Those that did not continued vomiting. So she did an open trial. We gave THC about 400 times. We had 100% blocking of vomiting and nausea. Unbelievable, because in clinical trials you don't see that. But we had complete blocking of nausea and vomiting at this stage, and of course she continued working on that. Unfortunately, we published that paper. It has not been taken over by pediatric oncologists, and it's a pity because it seems to work extremely well, and we can, once the treatment is completed, then they don't need it anymore. But there we stand. This is something that has happened quite a few times previously. Now, what about cannabidiol? Now, as I said, this is an interesting compound. It is non-toxic, causes no side effects. It can be given at huge amounts, and we see nothing at all. And we don't know how it works. As I said, here is the cannabidiol, and as I told you, it's almost equivalent in structure to THC, except that it is completely different than THC, and its actions are actually antagonize to some extent the effects of THC. And we initially worked on inflammation. We gave, in a model of inflammation to mice, we gave cannabidiol, and we saw that TNF, which is a compound produced in the body and has to do a lot with inflammation, the level goes down considerably in these animals. Oops. Here it goes. There are a few other effects that we have seen. And the reactive oxygen intermediates, I mean, oxygen that's produced in the body and is involved in many cases with the inflammation, also goes down. So once we saw that in animals, we did a trial in rheumatoid arthritis, and we found that when we give, in a model of rheumatoid arthritis, we give cannabidiol, a non-toxic compound, compound that causes no side effects. We gave it at 2.5 milligrams, nothing happened. We gave it at 5 milligrams. It was much more active, and I wouldn't go into the details of the tests, of course. This is 2.5. This is the 5 milligrams. But if we gave more than that, if we gave 10 milligrams or 20 milligrams, the effect was not so strong. So we have an effect which is typical for all cannabinoids, and in animals, I'm not so sure about humans, which is an inverse U type of effect. At a low dose, you don't get anything as expected. At a certain dose, one gets the effects that one hopes to get, and then it starts going down. This is very typical, and it is complicated, because if we see that in humans, we have to find the therapeutic window that one has to work on. Now, we also looked at, you see in the elbow, or during a rheumatoid arthritis, one sees a lot of this compound, the TNF, that has to do with the inflammation that's being produced. And if the animals get cannabidiol, CBD, then we see that the synovium in the elbows produces much less TNF. So here we see that it is an effect that is definitely worthwhile looking in humans, because we have the same effect in humans in rheumatoid arthritis. But to the best of my knowledge, it has not been tried any time in humans. Again, these compounds have not been tried in humans. Now, rheumatoid arthritis is an autoimmune disease, and so we decided to see whether cannabidiol works in other autoimmune diseases. Maybe it's the same mechanism. So we chose to look at diabetes type 1. Diabetes type 1 is an autoimmune disease, and there is a good model in animals. There is a strain of mice that have been treated so that all of them have diabetes type 1 disease at a certain point. So we looked at these animals. We waited until the 11th week when they start getting the effects of diabetes type 1, and then we started giving them cannabidiol. And indeed, while most, essentially all the animals that did not get CBD, cannabidiol, got diabetes type 1 and died as expected shortly, those that got CBD, only 30% of those, only 30% got the diabetes type 1. And I sincerely hope, that we should manage somehow or other give it to human patients, as it works so well, and the compound, the cannabidiol, is completely non-toxic. Well, in another case, just another one for those that may be interested, we gave these animals to the histologist, and he looked at various things, and he said, well, the one thing that I'm very much interested is, we look at the percent of intact cells, and those animals that didn't get the CBD, only 8% of these cells were in a good shape. When we gave them CBD, 77% were in a good shape. So chances are that we'll have the same thing in humans, but this has to be tested. Another point that I want to make is epilepsy. As you may have heard, children in the U.S., many other places, that have epileptic seizures 20, 30, 40 times a day. In many cases, nothing helps them. Parents have found that cannabis with a lot of cannabidiol does seem to help them. Well, unfortunately, nobody seems to have paid any attention to a paper we published 34 years ago, and it is still the only paper, a real clinical paper on the topic. We took 15 epileptic patients, not children, patients that did not benefit from anti-epileptic drugs. We gave them large amounts of CBD for quite a few months, and we had to prepare that from hashish that we got from the police, and contains 5% of cannabidiol, so we worked very hard to produce it. And we found that essentially the four patients out of 8 remained completely free of seizures. 3 patients had partial improvement, only 1 did not show any improvement. With the controls, we got essentially nothing. So we had strong data, and it should have been repeated since then. Now there is a lot of noise about it, not about the paper, about the treatment. There was even in the New York Times editorial about cannabidiol and epileptic children, but nothing has been published. People at the moment are trying to do clinical trials with cannabidiol in epileptic children. Well, we too are going ahead with that. It's a shame. It's a paper that was published 35 years ago. It should have been repeated. This was a very small scale. We couldn't do it on a large scale. These things are not so simple to do. Anyway, in anxiety, it certainly works in anxiety. It has been shown to work in anxiety in both mice and humans, and it has been found to be due to the synthesis of endocannabinoids. It has been found to work in schizophrenia in a clinical trial. In Germany, where it was possible to do a clinical trial, a German group took schizophrenic patients and gave them for about a couple of months, gave them cannabidiol, and compare the effects with the effects of a well-known drug, which is amisulpride, which is widely used, and they found that both drugs acted to the same extent, with one exception. The anti-schizophrenic drug that's being used has serious side effects. Cannabidiol did the same without any effects. So I sincerely hope that people will go ahead and use cannabidiol in schizophrenia. And there is a long list of other effects with cannabidiol. So one has to think, start making speculation of how come that such a compound has such a wide spectrum of activities. Is it possible that it is acting on some basic regulatory mechanism? Now, a basic regulatory mechanism is, for example, epigenetics. Those of you who may not be aware of that field, epigenetics has to do with changes in the DNA, but not changes in the basis of the DNA, of the compounds that form the DNA, but by making small chemical changes, by putting a metal group there, here, or an acetyl group there, slowing down the activity. So if there is a gene that acts over time and ultimately, through a series of reactions, produces a compound, at doses at too high level, then maybe by methylation one can block it down. Of course, this is well known. This is one of the bases of epigenetics. So is it possible that cannabidiol works an epigenetic mechanism by slowing down the overactivity of some genes? And then it can explain why it acts on so many things, because it acts on a basic mechanism of different diseases, a gene that possibly works over time. And indeed, a very good friend of mine has shown that this is true. They show that this is true in the treatment of psoriasis. It has been known for a few years that cannabidiol helps in psoriasis, which is due to the proliferation of certain skin cells, the keratinocytes. And indeed, they found the DNA methylates the gene that is involved in psoriasis, and it blocks the production of, apparently, of something along the way, and that's the way it acts. It blocks, it methylates the DNA. Is it possible that CBD actually is a regulatory compound acting on other DNAs? We don't know. This paper was published just a few months ago, and they're trying to look at others. We're trying to look at other cases, maybe in schizophrenia, maybe in other things. But the fact is that it does help. Now, my question is it possible that cannabidiol mimics the effect of something that we already have in the body and which regulates the effects of DNA? It doesn't make sense that we don't have a regulatory system in the body. Possibly DNA can be regulated, the activity of DNA can be regulated by endogenous compounds, and some of these compounds have been shown to act, and anandamide, as a matter of fact, also does that, but it has been shown in a single case. So, as cannabidiol is so active, we've tried to look into compounds that may be more active, and we have done a few syntheses, and this is, for example, this synthesis here, and we have looked at the anxiety that cannabidiol causes and whether our compound is more active. Yes, it is. Now, we looked at mice. Now, mice do not like to be in the limelight. They don't like to be celebrities. They don't like to be in the public eye, and if possible, they try to hide in some closed space. And this is kind of an anxiety. But if we give them cannabidiol, they move out for a longer period, not for a very long period, but they try to move out. They are less anxious, and we found that our compound is much less, it lowers anxiety to a much higher rate than CBD itself. So instead of being here in the dark, the mice go outside and look around what's happening. So let me tell you a few things about 2-AG. 2-AG is one of the endogenous compounds that our brains form. Now, 2-AG is an interesting compound. When there is a brain trauma, the 2-AG goes up, not in humans. Nobody knows whether that's true in humans, possibly yes. But in mice, when there is a brain trauma in mice, the concentration of 2-AG goes up. Now, we thought, well, maybe it goes up because the brain has been traumatized and therefore it does all kinds of strange things. Or maybe it's a protective mechanism. It's kind of a therapeutic mechanism, self-therapeutic mechanism. So we saw, first of all, that the 2-AG, the levels of 2-AG after brain trauma go up. Whoop, after 4 hours they go, this is the control, after 4 hours they go up to a very large extent, 10 times more. And they go on being high for many, many more hours, even days. So we decided to do something else. We synthesized 2-AG and gave the mice that had undergone brain trauma, gave synthetic 2-AG. And yes, indeed, the synthetic 2-AG helped with the brain trauma in a variety of ways. Again, I wouldn't go into the details of the experiment, but for example, walking on a narrow strip of wood, an animal will fall off after brain trauma, and in this case we see a much greater improvement, things of that sort. And yes, 2-AG definitely helps. It helps reduce the edema in the brain, something very typical for brain trauma. There is edema in the brain, very dangerous, as you, of course, well know. And this is something that we see by administration of 2-AG. So here we have a compound, an endogenous compound, that works as a protective mechanism, if you wish. And we gave the brains of these poor mice to our histologists, and indeed they found that when given 2-AG, the amount of damage, and this is the amount of damage, is about 50% the amount of damage seen in the animals that do not get the 2-AG. It should be tested in humans. Again, nothing has been done so far, unfortunately. Now, brain injury causes not only actions on the glutamate, cytokines, and so on, it also causes actions, other actions like vasoconstriction. And this vasoconstriction may lead to cerebral ischemia. And cerebral ischemia is something that's difficult to treat. We wanted to see whether 2-AG helps. We found that it helps to a certain extent, but not to a very large extent. But in the body, one sees considerable effects from endogenous compounds. So we thought maybe one of the many compounds that are formed there causes the vasodilation. Now, if I were in a company, would synthesize 1,000 compounds and see whether any one of them helps the vasoconstriction. Now, in an academic lab like mine, a small lab, we have to spend a little bit of time thinking. And my way of thinking is our body is stingy. If it can do something without spending too much energy, it will do it. And we've seen that in steroids. This is a steroid skeleton. If once the body has learned how to make a steroid, it will use the steroid skeleton to go ahead in varieties of effects. For example, changing here, one gets androgens and estrogens. Here, corticosteroids. Here, aldosterone. Here, others, progesterone, and so on. So we thought that maybe this is what's happening with our brain. We know how anandamide is formed. Don't go into the chemistry. We know that it goes through phosphatidyl ethanolamine, goes into anandamide. Now, phosphatidyl ethanolamine is always present in the body with phosphatidyl serine. Well, is it possible, is it just possible, that these two compounds have the same mechanism? The only difference here is a small one. Here. The same compound. So maybe if the body makes anandamide this way, maybe it will make the relevant compound by the same way. And we looked for this particular compound, which I just mentioned, arachidonylserine, and which is very close to anandamide. And yes, it was in the brain. We found it there. We synthesized it, and we looked at the damage that, whether it acts on the brain after damage and the vasoconstriction. And yes, we definitely saw very good results. As a matter of fact, this compound, which does not bind to the CB1 or CB2 receptor, is actually a compound that lowers vasoconstriction. So the body is stingy, and the body doesn't want to do novel things. It can take something that it knows how to do and just make a small change and get another compound which is just as active. And if we compare the effect of 2-AG and the effect of this compound, arachidonylserine, which we call ARAS, the two effects are the same. So we can expect that many of these compounds that we see, about 200 of them, have effects of this or other nature, and we have seen many other effects. I don't have the time to go into all of them. So chances are that we are looking at a system related to the endocannabinoid system, part of it, and it will act in many, many other ways and possibly in many other areas. Now, then we looked at the action of the CB2 receptor. Said that there are two receptors, CB1, which causes the high, and the CB2, which does not cause the high, but it causes all kinds of effects. And group at NIH with me, we made the list of everything that was published at that time, 2 years ago, on CB2, and we found that in a large number of diseases, we see enhancement of the endocannabinoid levels, we see expression of the CB2 receptor goes up, and we see the effects of specific endocannabinoid compounds that act only on the CB2. And here you can see, starting from heart problems, atherosclerosis, stroke, heart failure. These are all published papers, not by us, by many other groups. We see in autoimmune diseases, fatty disease, obesity, liver fibrosis, and so on, liver injury. We see all kinds of inflammations and pain and skin disorders and bone disorders. Surprisingly, it is a very efficient CB2 specific compounds are very efficient in osteoporosis. So, again, is it possible that the CB2 receptor, not so much the CB1 receptor, but the CB2 receptor is essentially part of it's part of a system that is involved in protection, the way we have the immune system. But the immune system basically guards us against foreign proteins. This is one of the main reasons we have an immune system. Without the immune system, we'll be dead probably within 24 hours. But there are many other things that are not directly involved with the immune system. Is it possible that 2AG works in parallel and together with the immune system in many other respects where the immune system is not directly involved, and we saw such a large number of effects? Quite possible. It has to be shown that this is correct, but it seems to be true because since we published that review on dozens of effects caused by the CB2, stimulation of the CB2 system, many additional papers have appeared in many, many different fields showing that the CB2 receptor system is involved. To what extent, we don't know, but it is involved. So I would like to summarize what I've been trying to say. The cannabidiol, the CBD, which has so many actions, may act through DNA methylation. It may work on regulation of the DNA, and by playing around with CBD, one can it can lead to a wide spectrum of novel drugs. We have synthesized several different cannabidiol derivatives, and they seem to do that. To what extent they will do that in humans, again, we don't know. The CB2 specific agonists, and one should it should be different. CBD does not act on CB2. CBD acts differently, but the CB2 specific agonists, again, work in parallel to the immune system on different diseases, not necessarily the same diseases, and it may be part of the general protective system. And the third point I try to make in a few words is those complexes of fatty acid bound to amino acids, of which we have 200 or 300 in the brain, may tell us a lot about the biological processes and to novel drugs. Now, is it possible that these 200 compounds work not only on, let's say, diseases that we know, but also on our psychological setup. We here have a group of 50 participants. I'm sure that each one of us has a different personality. We know nothing, absolutely nothing, why we have different personalities. Biologically, we don't know anything. Is it possible that here there are a few hundred compounds? It's a pure speculation. We have a few hundred compounds which seem to have activities. Is it possible that this play around with these compounds has something to do with our personalities? I wrote a review on that topic with a lot of references possibly doing that at the invitation of a psychology journal, and I'm not a psychologist, and I was sure that the editor will say go home. He said that was, I'm pretty happy with it, let's see what happens. So we published that, and the people have been interested. Maybe, just a pure speculation, it may have something to do. But the one thing that I didn't mention, and I want to do that, is that when I was introduced, we were working on PTSD, post-trauma, and there has been, the effects seem to be reasonably good with THC. We work on graft versus host disease, GVHD, prevention of GVHD, and it seems to be doing. We have published a little bit on that. I mean, obviously with clinical colleagues, I wouldn't know how to do it. And there is a lot of work on non-cancer pain, and my good friend Mary Lynch from Canada has reviewed all that work, and she found that of the 18 trials, 15 meet the inclusion of criteria, very strong criteria, and she found that cannabinoids, both THC and CBD, work on pain, not a very potent. They're modestly effective in neuropathic pain, but they have no side effects. So it is worthwhile looking at those. And I want to end by saying thank you to many of my collaborators, some of my students here, and many of my colleagues and all over Israel, and many of my friends abroad. You can see starting from the UK and going around many places in the world, a good time to visit almost all of them. The one that I have never visited is Dr. Maslov. Dr. Maslov organized a meeting in Siberia and in February. And he said that the weather was okay. He said it's minus 25. Usually they have minus 35. So I said, no, thank you, coming from a semi-tropical place. No, not really. So I thanked him, and I thank you.
Thank you for this wonderful and incredibly informative presentation. I'm so glad to see so many of my colleagues from anesthesia, pain specialists, pediatric pain specialists, psychiatry, so many different groups here to share this. As I mentioned at the very beginning, the goal of academic medicine is to bring people together and share these ideas, and you've given us so much to think about. I'm sure it's going to lead to numerous other studies and much work. If we could, we take a few minutes for questions, and then we can all...
If I can answer them.
I'm not a clinician. And then we can proceed. There's a luncheon outside. There's also a CME sign-up for those who would like it outside. And then we can come back in here, just informally spend some time together. We have about 2 hours, and there's a luncheon. So any questions? All that.
Yeah.
Any?
You mentioned about the cannabinoid on the neuropathic pain. Is it a mediated through the metabotropic glutamate receptors?
Sorry, my wife has to say. Yeah. Well, can you repeat that? I was...
Okay, the last line, one of the points you mentioned,
Yeah.
you know, the cannabinoid receptor system, they have something to do with the neuropathic pain. And I wonder, this kind of activity is a indirect, you know, action mediated through the mGlu receptors.
Actually, the mechanism is not known. I would assume that your suggestion is a correct one, but there is just not enough in the literature to say yes or no. I wish there was. Yeah.
In the animal study where they were given the cannabinoid receptor antagonist and they weren't suckling, you mentioned that in certain instances you were able to administer a dose of THC and it was able to counter the antagonist?
Yes. After the poor pups die after 6 or 7 days from hunger, they just don't suck, they don't get any milk. But if we give them large amount of THC or enough THC, it can kick out the antagonist from the receptor. And then there will be no receptor. There will be no antagonism. The receptor will start working again, and therefore they start sucking. Presumably that's what happens with humans. We know that THC or cannabis is useful. It enhances appetite. As a matter of fact, THC as a drug is allowed in the U.S. as a drug for enhancing appetite in HIV patients and some other cases, and it is being used. It enhances appetite to a certain extent. Surprisingly, there is something, and we talked about it just before, people that use cannabis in a few cases, they actually, an antagonism is formed and they start vomiting. In a few cases, a few percentages of those that they use it, but surprisingly, they start vomiting, have nausea. It seems to be some kind of an immune system, possibly the THC binding to a protein being acting on the immune system. Therefore, the immune system is an antagonist, and therefore people start vomiting. This is a new thing which has been seen several times, and the Mayo Clinic journal asked me to write about it. It seems to be, well, not prevalent, but there have been quite a few cases. Yes, sir.
So, thank you. First of all, it was a very interesting, wonderful talk.
Thank you.
Now, you demonstrated efficacy on many different disease states in a very impressive manner. And you've also speculated that there may be some epigenetic demethylation, and if it's acting on many of these different disease states, you would suspect that this demethylation is occurring at maybe several different.
In certain cases, not all of them.
So my concern is, and I'm always suspicious of any drug, is whenever you affect one mechanism, there always seems to be an alternate pathway. There are always side effects. And with all these different effects, good effects on different diseases, a possible general demethylation on different gene sites, it seems almost unfathomable to me that you wouldn't have a lot of other side effects. And I don't know if, in the long term, something's going to show up that we're missing, but it almost sounds too good to be true from the perspective of having so many good effects with no bad effects.
Well, you reflect exactly what I've been thinking about. I mean, how come that cannabidiol affects so many disease states and does not show any side effects? It's unbelievable. It doesn't. I haven't seen it with any drug that I know about. It should have side effects, but it doesn't. Now, I don't know why. Maybe it is specific. Maybe it's a specific methylating agent when a certain DNA acts strongly, not all of them. Maybe it acts that way only in a few cases. But the fact that we know it does not cause side effects, and it has been tested for toxicity. NIH did a very thorough 400-page report on toxicity of CBD. NIH wanted to know whether people that are smoking cannabis are doing something to their body because they smoke cannabidiol as well. And they prepared a big report. As a matter of fact, just in parentheses, that was done 25 years ago. And then NIH lost the report. And I got a letter a few months ago. Do you have a copy? I said, yes. Please send us a copy of our report. And so I had an opportunity to read the report. Yes, it says it's not toxic. So I don't know. What you're saying, as I said, reflects my way of thinking. It should have side effects, but we haven't seen them. So I don't know where we stand, but from the few clinical experiments done, we see no side effects.
Did you not say that it's possible it's working indirectly through an andamide? And possibly that's one reason why it's specifically to certain genes and not others.
Yeah, one of the things that cannabidiol does, at least that has been seen in schizophrenic patients, it enhances the formation of anandamide. So maybe the major mechanism is not a methylation. Maybe the major mechanism is enhancement of anandamide production. I mean, we are at a stage where guesses and facts have to be put together. Cannabidiol definitely enhances the formation of anandamide. Maybe methylation was just in this particular skin disease case.
Perhaps should we break here and then have lunch? Because that appetite slide is having an effect on me, so I think maybe we should have lunch at this
Thank
point.
you.
And then we'll have time to come back in and regroup and sit informally and spend some time together.
And before we go, I would like to thank the Kessler Foundation for inviting me, being able to present some of our results here. And I hope that we shall have the opportunity to work with the Kessler Foundation and do something in the field. The one thing that we're trying at the moment to do is to try cannabidiol on cases of severe anxiety, very severe anxiety in patients that want to commit suicide, and trying to see whether it will act. We have administrative problems at the moment getting it approved, but I hope that we shall be able to do it. Thank you.